All right. Well, on behalf of the Boyce Thompson Institute and our Centennial Planning Committee, I would like to welcome you all here to our 2024 Flash Science event. Thank you for being here. For those of you who don't know me, I'm Delaney Sickler. I work in education and outreach, and together with my colleague Bob Kohut, emeritus faculty here at the Institute, we are delighted to bring you this program tonight. So, for those of you who don't know, I think you might all know, but we are celebrating our centennial anniversary this year. We are turning 100 years old on September 24th, and we have a bunch of events that have been ongoing this year and that continue through October. So, grab a flyer if you don't know about them or connect with us online. Before we begin, I just want to start by thanking the folks that are joining us virtually. Any of the students who are from Future Farmers of America groups or 4-H groups or the Science and Technology Entry Point program, thank you for joining us virtually, as well as the Scientific Advisory Board members. I'd like to also thank our judges, Jillian Lindstrom from Brandmint, Benjamin Strabler from he's a Constituent Services Representative from Mark Molinalo's office, Alex Hagan from Cornell's Center for Regional Economic Advancement, and we have Shane Way and Sophie Way from Ithaca High School. Thank you. And lastly, I want to thank all of our presenters. It is not easy to take what you do and translate it to a general audience, especially when you're experts in an area that is complicated and has a lot of research results. And the work that you've done over the last week, 2 weeks to perfect your talk and to practice and put energy in is much appreciated. So I hope that you can take this scientific communication skill into whatever you do next. And lastly, for the audience, I hope that these 3-minute talks are the start of something, maybe a new conversation, new interest, and maybe ignite some curiosity about the world around you. So thank you for being here. And I want to hand it off to Bob Kohut. Bob is our MC for the evening. This is Bob's 4th Flash Science, and we couldn't have done it without him. So thank you, Bob.
Welcome you all here. This is the 4th time we've gone through Flash Science here. But let me just give you a little bit of background. Flash Science started at the University of Queensland in Australia. And at that university, as I understand it, it worked its way up from the programs to the departments to the college. And then there was a competition at the top of the university. So it was an all university kind of effort there. I read about this and then I thought, gee, it might be nice to try this at the Institute. So we did, and it was very successful. People enjoyed it. It is a challenge. And essentially, what the challenge is to take complicated science and present it to the public in such a way that you do not use any scientific jargon. That is really essential for science to make that transition from the lab into the community, if you will. And so it's hard to do, but it's essential in today's world that it be done. And when I describe what this is like, I kind of think of one of the scientists here being at home for Thanksgiving, and Uncle Joe asks him or her, tell me about what you're doing in your research. Now, Uncle Joe understands a little bit about science, but he's clearly not a scientist. And so your challenge is to explain to him what you are doing so that when you finish your little discussion with him, he walks away saying, I understand that. And the challenge is to do it in 3 minutes because the turkey is going to be on the table. So you don't have a lot of time to do this. And that's sort of where we are here today. We've got 10 contestants who are going to be given 3 minutes each to tell you about their research without using any jargon or technical terms or any visual aids either. So it is challenging, but it's fun. It's fun for them, I hope, and I hope it's also fun for you so that when you leave here, you will have a little bit of an understanding of what kind of research is going on here at the Institute. All of the people you're going to hear today are very well qualified to explain their research. They're challenged to explain their research, not using jargon. Okay? All right. So with that, oh, I will also say that the judges are going to have a difficult challenge for them to decide who here in the top 1, 2, and 3, the person who ends up in first place will receive $500. So it's worth the effort. Okay, first up is Tanner Robinson. Went to Utah State for bachelor's and master's and PhD at Cornell. He works in the Feiwei lab here. The scientific title that he would use if he was going to be giving you the jargon-filled version of it is C-terminal extension in hornwort RBCS mediates paranoid phase separation in hornworts. You got that? Okay. His non-technical title, Holding Hands in the Dark Reactions, How Hornworts Stick Together to Make Their Photosynthesis Better. So that's pretty straightforward. We've asked each speaker to tell us 1, 2, or 3 things about them that many people here do not know, but might be of interest. A big fan of Pride and Prejudice, especially the Keira Knightley version. Plays the banjo and learned to solve. This tells you a lot about this guy. Learned to solve the Rubik's Cube as a team because he thought it would help him get a girl's attention at church camp.
It didn't.
He also says it didn't work.
Okay. So photosynthesis seems like alchemy. Plants take light and they take air and they turn that into the sugars that drive all of life on Earth. But there's a flaw at the heart of photosynthesis. Rubisco is the protein that's responsible for taking CO2 out of the air and turning that into sugar. But Rubisco can't tell the difference, well, it has a hard time telling the difference between oxygen and carbon dioxide. And when Rubisco reacts with oxygen instead of carbon dioxide, the plant has to expend energy in order to reverse that reaction. Some plants have gotten around the limitations of Rubisco by putting all of their Rubisco into a special compartment. And then they concentrate CO2 into that compartment. And this reduces the likelihood that Rubisco will react with oxygen and instead with CO2. And this greatly enhances photosynthesis. Models estimate that by putting this sort of structure into a crop plant, you can increase yield by up to 40%. And hornworts, which is the organism that I study, they're the only land plant to have this kind of Rubisco compartment. And they're kind of humble little plants, and I think greatly overlooked. And so the goal of my PhD was to study how they make these structures and how they can enhance their photosynthesis. And to start, we drew from algae because this structure in hornworts is a lot like a structure found in algae. And in algae, they have a special kind of linker protein that it acts like a magnet for rubisco, and it pulls all the rubisco together into one kind of one place. And so I went looking for a linker like this in hornworts, and I couldn't find anything, which was at first frustrating. But then I took a closer look at the hornworts rubisco itself, and I found that it has like additional sequence at the end of the protein, and it sort of looks like it hangs out of the protein like an arm, reaching out. And when you cut that arm off, and when I cut that arm off and fused it with a protein that lights up under a microscope, you can see that just that arm part is enough to light up the structures in hornworts. And so this made us think that this structure, this extra arm, is maybe what's responsible for forming these structures in hornworts. So when we took just this hornwort rubisco and then expressed it, we put it into a plant which doesn't have this structure at all, it also formed these structures. So we think what's happening is that the hornworts rubisco are kind of reaching out, and they're grabbing their neighbor's hand, and they're holding hands. And so we're excited about that.
Great presentation. Is the hope that these lessons could be learned and applied to maybe crops or other applications to try to improve the yield?
Yeah, exactly. Yeah. So yeah, like I said, estimates are, up to 40% increased yield if we can integrate these structures into a crop plant. So the upside is quite large, I think. Yeah. Yeah.
The CO2 in the air keeps going up. What kind of effect would that have? Wouldn't that help increase productivity in plants?
Yeah, I think it will. Yes. I think these structures concentrate CO2 so much around rubisco, like I think 50 times atmospheric. So the CO2 in the atmosphere would have to get quite high for this structure to not be as advantageous.
Is there a reason why hornworts are the only one?
Yeah, that's the golden question. No, maybe I don't. Well, certainly there is a reason, but I don't think we know that reason yet. Yeah.
Our next presenter is Shuibo Shou. Did her undergraduate studies at, or rather a PhD, at the Institute of Genetics and Developmental Biology, the Chinese Academy of Sciences. She works in the Changchun Fei Lab. The scientific title of her presentation would be The Demographic History and Genomic Diversification of Bottle Gourd. And her flash title is The Fantastic Journey of the Bottle Gourd Across Oceans and Time.
Hello, everyone. Today, we are going on an exciting journey with the bottle gourd around the world. Do you know the bottle gourd is one of the most ancient domesticated plants? It has been cultivated for around 1,200 years. People have used it for all sorts of things like making pots, food, musical instruments, and even medicine. But here is a big mystery. How did the bottle gourd spread across the world? Now, imagine us as activists exploring the evolutionary history of the bottle gourd by looking at the genomic information of 200 different kinds of bottle gourd from all over the world. We found the bottle gourd originally grown as wild plants in South Africa. Then they traveled alongside humans, reaching Europe around 12,000 years ago, and then to East Asia around 7,500 years ago. More interestingly, we found the bottle gourd traveled from the Africa to America by floating across the ocean currents around 10,000 years ago, becoming the only plants with global distribution pre-Columbia times. Bottle gourd plays the crucial role in the early human society, especially during the early days of farming, serving as containers and food. However, as people developed the pottery, porcelain, and ironware for making containers and tools, the bottle gourd become less important. So we have found the shrinking habitats of the bottle gourd and the decline of the bottle gourd population as a result. This adventure is not just a story, it's a lesson in how plants adapt and change, which can help us growing healthier crops. Nowadays, the resilience of the bottle gourd makes it an ideal rootstock for other plants, including the watermelon, help them to resist disease. Understanding the rise and fall of the bottle gourd can also help us develop the better strategy to when facing the challenges such as the climate change and emerging plant disease. Thanks for joining me on this amazing journey. Questions?
You mentioned they were domesticated 1,200 years ago, right?
Yeah.
12,000. What is 12,000? Oh, 12,000. So it was before the spread.
Yeah.
Right?
Are they in Australia, or is there a reason that they're not in Australia too?
We didn't collected the samples in the Australia, so we cannot use the population genetics analysis to analyze how when arrived at the Australia.
Does the dash circle?
Oh yeah. The dash circle means the gene flow. The gene flow, the main route of the bottle gourd from the Asia is from the first is from the West Asia and then to East Asia. But there is also a gene flow from the Africa to South Asia by the floating from the Indian Oceans. Yeah.
I think a similar question about the transit across the Atlantic. And I'm wondering, is this one of the things that Darwin was looking at in terms of seed dispersal in bottle gourd, or is this a different species? And is there any sort of ecological data to show the feasibility of that transit?
Yeah. Do you mean
so the 10,000 years ago is a reconstruction based on genetics and genomics, right? And so I'm wondering, is there some real world observations about the transport of these type of plants floating that distance?
How long does a bottle gourd survive if you dump it in the ocean?
Yeah.
Next up, Evelyn Elkington did her undergraduate work at the University of Texas at El Paso. Works in the lab of Julian Fay on a project involving hybrid vigor And so her presentation is going to be from the lab to the farm and putting results into practice. Oh, up. Hold it. Sorry. She did submit 3 things about herself that are kind of interesting. She loves alligators, would like to meet one someday. She learned to do trapeze tricks from circus performers when she was 12. She can do a cartwheel, but only one.
So did anyone notice that this past winter was pretty mild? Like we did not get a lot of snow. Or so I'm told, because I'm from Texas, but the last summer, the entire month of July, the peak temperature of the day did not get below 100 degrees. It was really, really hot. And because it was so hot, there wasn't a lot of water for agricultural or crop purposes, which is why my team is working on making one plant make more food. Because if we can put our resources and our energy into a smaller amount of plants and make more food, we can start to create more sustainable agricultural practices. So right now the project we're working on is called the Hybrid Vigor Project, because it was found by a researcher in Brazil that when you put 2 specific types of tomato plants together, the resulting child plant is way bigger and can produce about 3 times more tomato than either of its parents. And as it turns out, that change is due to a certain region of the plant's DNA. And so what we're working on now is finding what changes can we make to the DNA that will allow us to take that change and put it into other types of plants, like maybe peppers, potatoes, things like this. So that all sounds good, but we are kind of left with one problem, which is how do we take this research that we're working on in the lab and actually put it into the field? So that's kind of where the translational part of translational science comes in. Like, for example, right now the tomatoes we're working on are specifically lab tomatoes. They don't taste very good. The plants grow differently than they would in your garden or in the field. And so we're also working on taking what we know about these lab tomatoes and putting it into commercial varieties that farmers can actually grow. So kind of the broader impact is being able to have these results go directly from what we're working on in the lab to commercial varieties that farmers use, is we can have a direct result that is very easy to see the impacts of in the grocery store. When you buy tomatoes, they're cheaper, they're easier to find. And eventually we want to also be able to share these seeds or share how to make these edits with farmers in other countries who may be experiencing widespread food shortages. So in theory, that gives more food to go around and kind of helps have access to fresh fruits and vegetables for everybody. Translational science has a ton of different applications, and we're always working on things from getting them from the lab into the real world. So if you have any questions about any of the projects we're working on, let me know. Thank you. So does anyone have any questions? Did I explain it like so well? No one knows? Or they're confused? What's up?
Are the tomatoes, were they all broken around at the same time?
Yes.
So that's the result then after a set amount of time?
Right, yeah. So they were all planted at the same time, like all the seeds were in the dirt at the same time. And then once they reach maturity and start producing fruit, and the fruit also matures and becomes red, like when you would eat it, that's when they were harvested. Mm-hmm. Yes.
So besides increases in productivity, are there any other aspects of hybrid vigor, increases in disease, insect resistance?
So there are not any increases. There's about the same amount of susceptibility to these types of problems. Mm-hmm. Yes.
Do plants make more tomatoes and so like nutrient content matters? Is the tomato basically the same as the others, or the same tomato that is making?
So that's the problem with using lab tomatoes is the nutrient content doesn't really change, but we don't know how it will change. This editing this specific part will change the taste or really anything else about like the actual properties of the tomato, because we're using just the lab tomatoes right now. That's kind of a later, when we're testing it in commercial varieties, we will make sure that those things are the same and that it tastes good so that it can actually be applicable to real world. Mhm.
Next speaker is Yana Campos-Brazieri. Did his undergraduate work at the Federal Fluminense University. Works in a lab of Fei Wei Li. The scientific title of his presentation would be Understanding the Chromosome Hoarding Syndrome of Homosporous Ferns. The flash science title is Why Do Ferns Hoard Chromosomes? Now, two interesting things about it. He's the main organizer of the 2024 International Youth East Meets Yeast Eat Great Gun Lean Labs Bake Off. Now, there's a mouthful there, but the bake off part got my attention. Okay. And the last one here was really a surprise to me. He's not a natural blonde.
So ferns, they are plants that don't produce any seeds, and that you can see they come in very different shapes and sizes. And what's interesting about them is that they have a lot of DNA content. They have, to give some perspective, they have 100 times more DNA than tomato or orange trees do. And today, until this day, we don't actually know why they have accumulated so much information in their cells. One of the ideas that we have is that there's a relationship between the way ferns reproduce and this hoarding syndrome that they have. So ferns, during a stage of their lives, they have the female and the male reproductive structures very close to each other. And what can happen in a very lonely day in the midst of the forest grounds, they can actually self-fertilize. So this new fern baby has all of the DNA coming from a single source, which makes the DNA very similar. And the problem with that is that if there's a change in the environment or a new bug comes and starts eating these plants, it's less likely that they will survive. To cope with that, one thing is to have more DNA. So that's not very intuitive, but picture this. If you have 40 chromosomes and you duplicate all of them, you have a total of 80. That's not exactly going to solve the problem, but it's going to create a resource for changes to happen in the sequences. And by accumulating changes in the DNA sequences through millions and millions and millions of years, the chromosomes start diverging and they're not so alike anymore. This question of why ferns hoard so much DNA has been bothering researchers for decades now. And it's still very interesting to a lot of evolutionary biologists, like some of us that are in the Lee Lab here at BTI. To actually answer this question, what we have to do is to sequence the DNA of all of these plants, but that's still very costly. We do hope, though, that in one day we can actually, as DNA sequencing gets cheaper, maybe collaborating with other institutions or having new students that are interested in working with us, we will actually be able to solve this problem and understand why ferns hoard so much DNA. Thank you.
So there are some for various little genome.
Yes, yes. But it's a minority. So ferns it's around 11,000 species, if I'm not mistaken. And there's 100 that have the smaller genomes, yeah. They have a different reproductive mode. The male and the female don't get close together. Separate, yeah.
Are these, all of these have the similar habitat, they live under similar environment?
No, so these, the one on the right is a land plant, right? It lives in the land, and the two on the left are aquatic ferns. The middle one is kind of like a swampy kind of vibe, and a little down to the water. And the one on the left, it likes to be soaked in water, but not like deep down in water. It's just like the moist, so moist. Yeah.
Oh, come on.
Whoa, yeah. Yeah, it's not a grass, it's still unfurling. We're not gonna go crazy here. Yeah.
Next speaker is Laura Kaminsky. She got her bachelor's in science in environmental science and sustainability at Cornell, and a PhD in environmental microbiology at Penn State, Go State. She works with Maria Harrison here. And the scientific title of her project would be the ecological role of myxobacteria in the hyposphere of arbuscular mycorrhizal fungi. The flash title science of that is predatory bacteria, friend or foe of fungal plant partners. Three things that she wants to share with us is she climbed into the canopy of a giant sequoia tree once. She owns a rescued, retired racing greyhound named Winnie. In her spare time, if she has any, she thinks up fun names for her future chicken flock. Some samples are Chicky Minaj, Edgar Allan Poe, obviously a rooster, and Ken Stefani.
In my research, I'm constantly thinking about relationships, particularly the relationships between plant roots living underground in the soil and the tiny microorganisms that live in that soil too. Just like human relationships, these range from positive to neutral to negative. Think of it like a neighborhood. There are some neighbors you like and hang out with, some you mostly don't see, and some that make your life really annoying. So these microbe neighbors can really help or hinder plant growth, and the more we understand them, the more we can promote the positive relationships that support plant growth and productivity without needing to rely on environmentally damaging fertilizers and pesticides. So with that in mind, I want to tell you the story of two different soil microbes today, one whose role we understand pretty well, and one we don't, which I'm studying. So our first microbe is a type of fungi called mycorrhizal fungi, and this forms positive relationships with plants. So the plant gives food to the fungi in the form of carbon, and this fuels the growth of fungal threads far out into the soil. The fungi, in exchange, scavenges nutrients from that faraway soil and brings it back to trade with the plant. Now imagine that you are a hungry soil bacteria living in that faraway soil. Think of this like a neighborhood where nobody has had anything to eat since breakfast. When a fungal thread grows into your neighborhood, it has food to spare, given to it by the plant partner, so think of it like an ice cream truck. It is hoping to trade this food with bacteria that will help it mine nutrients to bring back to the plant, and these positive relationships do happen, but at the same time, lots of other microbes are drawn to the fungal thread because of the food. One such group are a type of predatory bacteria called myxobacteria. That's the second microbe in our story. These bacteria attack and kill other microbes in order to steal their nutrients and support their own growth. So what are these predators doing around our fungal threads? We could imagine they're acting like a bunch of bullies. When the fungal ice cream truck drives into the neighborhood, they attack it and they steal the ice cream for themselves. Or they could be acting more like police, catching and punishing other bacteria that are stealing from the fungal ice cream truck. So I have a couple of these predatory bacteria that I've caught in the lab off of fungal thread samples. And now that I have them, we can start to do things like look into their DNA and see if they have the machinery to take apart fungal threads. We can also look at them under the microscope and see if they eat fungal threads that I offer to them or if they prefer bacterial prey. We can also grow plants with our fungal partner and then add in the predatory bacteria and see if this decreases the abundance of the fungi in the soil and impacts plant growth. So with these future experiments, we should be able to tell if these predatory bacteria are friends or foes of our fungal partner and therefore of the plant. Thank you.
questions?
Yeah.
Great. How many bacterial species do you think there are associated with this? Like, is it tough to get an idea from contamination?
So we know most of what we know from DNA inventories. So we'll take samples of these fungal threads and use a DNA-based method to get a list of all the different species present. So there's tons of other stuff besides just the predatory bacteria. Within the predatory bacteria, there's two groups specifically, a genus and a family that we see popping up consistently, which are actually different from the ones that I've caught in the lab, because that's a whole other conversation about how we catch these predators is kind of difficult. But yeah, we do have a sense of all the different species present on the fungal threads, and it's a lot. Yeah. Yeah.
Or
I can yell.
Yeah.
Okay. Are these bacteria specific to some special plant species, or is very universal with the host? Okay, any host.
The fungi specifically, or the bacteria?
The bacteria, your function.
So no, they are just sort of a general group that exists in soil, and they also congregate to plant roots, and then we're also finding them around the fungal threads, but they're also just kind of roaming the soil. So no, they don't have any specificity to, at least that I know of, any particular specificity to certain plant species or certain fungal species for that matter.
I think you just answered my question. How many, the ones you're looking at, can live on their own, and how much are dependent on growing the fungi?
So they can live on their, they need a prey of some kind. So when we catch them in the lab, what we do is you'll streak like E. coli on a petri dish that has basically no other nutrients in it. So then you'll place your fungal or soil sample, and the predatory bacteria will start eating down your streak of E. coli. And if you don't have some prey for them, it's harder to distinguish them from other bacteria in your sample. So they do need something to eat. But I think some of them can sort of switch lifestyles. They can be predators sometimes and survive, or be dormant otherwise if there's no prey around.
Next, Hongki's son got his bachelor's and master's degree at the Beijing University of Agriculture in vegetable science, works in the Zhangfei lab. The scientific title would be Construction and Analysis of the Graph-Based Pangenome of the Citrullus genus. Flash science title, Harnessing Complete Genetic Blueprint of Cultivated and Wild Watermelon to Accelerate Breeding. Some things of interest about him. He likes to read science fiction novels, and two of his favorites are Ender's Game and The Three-Body Problem. The sci-fi novels expand his imagination. It's interesting to explore complex ideas through fiction.
So yeah, watermelons have changed significantly from their wild ancestors since 4,000 years ago to meet human needs. Today's watermelons are sweeter, larger, and will come in various of colors and shapes. However, while they were bred for these desirable traits, they have lost the disease resistances that are found in the wild watermelons. This can be a common trade-off during domestication, where humans select the crops for specific traits. The phenotypic variations among watermelons can be due to the differences in their genetic materials. And understanding these genetic differences can help us develop better varieties more efficiently. A genome is a complete genetic blueprint, which can be referenced to study the genetic control of a trait. And since the first release of the since the release of the first watermelon genome in 2012, we have gained a substantial understanding of this knowledge. So, however, only a few watermelon genomes are available in public that are not enough to capture the entire genetic diversity. To fill this gap, we developed more than 100 watermelon genomes from both cultivated and wild watermelons. Yeah, and well, by using these genomes as the reference, we are able to further study the genetic variations among about 900 watermelons. Okay. Yeah. Well, from our study, although it is still ongoing, we have discovered genes that have been uniquely duplicated within the cultivated watermelons. And these genes might likely contribute to the cultivated watermelons' increased sweetness. And we know that American type watermelons are elongated, and this shape can make them easy to be easier to be stored and transported. And this unique shape is caused by a genetic change that are recently arised from the cultivars. That means they occurred after the domestication of watermelon. And we also identified the genes that are present in the wild watermelons that are responsible to the disease resistance, but completely lost from the cultivated watermelons. These can be the target for the disease resistance breeding brought from the wild watermelons to the cultivars. And by understanding these genetic factors, breeders will be able to provide better varieties and make watermelons more resilient, faster. And this can not only improve the watermelons we eat, but also support the global agriculture by maintaining the health and sustainability of this fruit. That's all. Thank you.
Hi, great talk. Is there any trade-off between disease resistance and then some of the traits that breeders have been trying to breed for, like sweetness or sugar content? Like, is there any relationship, do you think, or phenotype?
I can say there are, but I can say there are, but there can be. For example, if you have a disease resistance, like very close to a beneficial gene for the sweetness, but these two genes are so close, so they are linked together and segregated together. But the one in one individual, these two genes are one beneficial for the disease resistance, one harmful for the sweetness, and in the other individual they are reversed. So it's hard for us to break the link between these two to make a both sweet and disease resistant cultivars. Yeah.
Next up, Trevor Tivy. He did his undergraduate, or I'm not certain exactly what it was, at Oregon State University. Okay. He works in the Maria Harrison lab. Scientific title would be Spatially Resolving the Cellular Landscape of Orbicular Mycorrhizal Symbiosis with Metacargo tricantula and Rhizophagus irregularis. The title of the flash science presentation is Mapping the Landscape of Plant and Fungal Symbiosis. He tells me that he's been told he jumps like a frog when playing volleyball and runs like a Tyrannosaurus Rex when playing soccer. And that
great coaches.
And that just last week he saw his first scarlet tanager.
So when I was young, my sister and I created a map of our neighborhood. not the streets, but the secret paths that exist in the forest behind and between the houses. It made me feel like we knew something the rest of the world didn't. And we ran up and down these paths, communicating on these janky walkie-talkies and just exploring what was happening. Now that I'm a biologist, I make maps on a different scale. I make maps inside organisms, and I look where microbes are and what they're doing. So here at BTI, I study the interactions that happen underground, where plant roots meet soil fungi. In this relationship, plants give the energy they get from the sun to the fungi that live inside plant cells. In return, the fungus gets the plant's minerals they get from the ground, using their vast network of finger-like hyphae, and in doing so, acts like a natural fertilizer. In my map making here at BTI, I've made not one, but two types of maps. One map was of the plant root itself. With a fungus inside of it, I can see how each space contains their own collection of messages, like tiny forums side by side. For my other map, I chopped up the plant root to get the plant and fungal cells and look at these very same messages from every cell separately, like the survey was sent out to each one. These two maps, overlaid atop each other, provide enhanced resolution like you would find in a brand new computer monitor, or like an episode of CSI. They help me find big differences in parts of the fungus that looked identical to each other. They help me better understand the communication between plants and fungi, as well as communication between the plant cells themselves. And these maps I've created provide important information for future plant fungal explorers. These partnerships, known as arbuscular mycorrhizae, are found in the roots of many plants and many of our crops. Our maps can identify ways to change the plant, or in the future, the fungus, so that we can take advantage of the beneficial interactions that occur all around us. When I now go back to my parents' house, these paths I once mapped are gone. New houses have gone up, trees have been cleared, and nature has covered traces of walkways that we meticulously pruned from briars as children. But now, despite these woods being slightly smaller, I can appreciate them with a different sense of wonder, because each plant has its own map just waiting to be explored. Thank you. Questions?
Great talk. I like the analogy to like mapping with your sister. It's awesome. I guess what I'm curious about is like understanding kind of the messages that the fungi and the plant are communicating in the two species that you're looking at, and like, is that very species specific, those messages, or how would it work in like engineering that to something new and something different to incorporate a new plant or a new fungi?
Yeah. That's a great question. I think it really depends on the species. Some species are a little more cosmopolitan, I would say, and these interactions happen in like 70% of land plants. But there are definitely, different types of plants that can't benefit from this. And so trying to figure out ways that we can engineer plants to accommodate that, or find ways to, figure out ways to keep that fungi and maybe retain it a little bit longer would be, or in less restrictive environments would be useful. Yeah.
The one in the maps is it like a spatial transcriptomic?
Yeah. So that's like the size of, well, it's like the size of that. But the roots on the top are just mock. So they don't have anything in them. The roots on the bottom show exactly where the fungi is and what type of fungal structures. They're just colors, but those are the different structures.
Do we know of ways to promote mycorrhizal growth to benefit, say, crop production?
Yeah. So, I mean, that's sort of where actually Laura's stuff comes in too, because they're part of a community in general. And so just ways to promote that soil includes making sure that soil has a good community, that we're not nuking with fertilizers and things like that. Or what? Or tilling, right? Yeah. So it can be difficult. This interaction sometimes only happens when there's a need for the nutrients driving from the fungus to the plant. And if that's need's not there, then it's not going to happen.
Anna Hermans is next. I got her bachelor's degree from Heinrich Heine University in Dusseldorf, master's degree from the University of Bonn. She works in the Jim Giovannoni lab. Her scientific title would be The Confluence of Structural Variants, Whole Genome Expression Profiles, and Nutrient Accumulation in a Tomato Diversity Panel. The flash science equivalent is analysis of diverse tomatoes from across biological scales. Two things of interest: her younger sister is graduating from high school this summer, and she was a unicyclist in a circus.
Well, who of you had tomato in your food today at some point? I definitely had. Yeah, probably a lot of you. You may have not even noticed that you had tomatoes in your food. I'm a researcher here at BTI, and I'm really interested in understanding more of the biology that underlies fruit ripening and nutrition, especially in a crop that is needed for us, for our health, and needed in our diet. And tomatoes, even though a lot of people will refer to them as vegetables, are actually fruit, and they definitely are important. They're used in a lot of the foods that we eat during the day or on a daily basis. Tomatoes are also really easy to grow. I would assume a lot of you have probably been growing tomatoes at home. And they're also a lot of fun to work with because they have a lot of different phenotypes. And with phenotypes, I mean the visual appearance of the fruit, whether it is the shape or the size or the color of the fruit. And at BTI, I am characterizing 200 different tomato varieties. And I'm doing that at different biological scales. So that means I'm looking at, or I'm characterizing the visual appearance of the fruit, like I just mentioned, but I'm also looking at the genetic variability between those different varieties. So I'm looking at how the genome differs from one variety to another. And all that information is really helpful for researchers as well as for breeders to understand what kind of areas of the genome correlate or have an effect on specific characteristics of those tomato fruit. So for example, they can use this information for breeding of specific tomatoes. And those 200 tomato varieties, they kind of overlap all kinds of different groups of tomatoes. Some of them are commercial, so you can find them at the supermarket. Some of them are like lab tomatoes, so they have been researched a lot. And then others are wild tomato relatives. They are you can find them naturally in the Galapagos Islands. So what I do, I grow those tomatoes out in the field in the greenhouse. Then I harvest the tomatoes, and during the harvest process, I measure their weight, their size, and I take photos for color extractions. And then I freeze and grind the tomatoes, which I actually do with a coffee grinder. And I can extract nutrients and the DNA and the RNA. And the nutrients tell me how nutritionally valuable a tomato is, and then the DNA and the RNA tell me the genetic composition of those varieties. And then with this data, I've actually found an area in the genome that associates with a specific nutrient that hasn't been known before. That is great. And we're also looking at how the color we can use the color of the fruit to predict the nutritional value. So with that, I hope you all got a little bit of an understanding of how such a resource is developed here at BTI. Thank you. Yes.
You talked about we're looking at nutrients. What are the main nutrients that we receive from the tomato, and what nutrients would you like to see be more available, or even would you consider putting in things that nutrients like more protein, because I never considered the tomato as a protein source.
Yeah, so tomatoes, they mostly have carotenoids, which give the tomatoes the yellow to red color. And they also have flavonoids. They're pretty much colorless. And then sometimes they have anthocyanins. They would give the tomatoes the purple color. And then, so I don't know if many of you have heard of the Norfolk tomato that came out as a purple tomato, where engineers have actually included more anthocyanins into the tomato, so they're completely purple. I think that's pretty interesting. It is a GMO tomato, but it is really valuable in a nutritional sense. And then someone else in our lab is working on folate. It's a vitamin to introduce more of that into tomatoes. Tomatoes so far don't have too much folate, but we're trying to improve the nutritional content in that sense. Yeah.
Like nutritional differences between like the more specialized kinds of tomatoes versus like a, I don't know, like an heirloom tomato or something like that?
Yeah. I haven't, well, the yellow ones actually have less lycopene, which is one of the major nutrients in a tomato. So actually the yellow ones have a little less nutrition than the red ones. Some of the pimpinellifolium, so the small red tomatoes, they are very dense in their nutrition because they're smaller. But yeah, it's yeah, that's as much as I can tell you at this point. Yeah. Yeah.
Do any of the tomato species have a similar level of spicy compounds analogous to a pepper?
No, but it would not as far as I know. But it would be great to breed a spicy tomato. And pepper and tomato are fairly closely related, so maybe there's something we can breed into the tomato. Yeah. Okay. Well, thank you very much.
Next up is Sarah Gartner. She did her undergraduate work at Franklin and Marshall College, and here she works in the lab with David Stern and Amber Cotto. The scientific title would be Understanding C4 Carbon Capture in Ceteria fideris. The flash science title, The Story Inside a Plant's Kitchen. She indicates that she was a competitive swimmer for 15 years, will do almost anything for free food, and her favorite bird is a flamingo.
Good evening, everyone. As we all know, climate change is a big problem because the Earth is getting warmer every year. The global population is also growing, so eventually we will need more food to feed everyone. This is a major problem in the plant world because at these higher temperatures, many plants grow poorly, so they produce less food. One of the reasons for this is because photosynthesis is a temperature-sensitive process. But how does photosynthesis really work? That's the focus of my research. Specifically, I want to understand more about the machinery involved in photosynthesis. One of the molecules that I work on is known as rubisco. And in photosynthesis, rubisco is responsible for changing carbon dioxide into sugars. And without rubisco, a plant would simply not survive. But rubisco does not work alone. This is where one of the other molecules that I work on comes in, known as rubisco activase, or RCA for short, which was appropriately named because its function is to activate rubisco. Now, RCA exists in two forms, known as alpha and beta. The beta form is always present. However, the alpha form is typically only present when the plant is stressed, and that includes being at these higher temperatures. Now, let's picture this system as a kitchen or a bakery. And in this kitchen, rubisco would be our mixer, combining all of our essential ingredients together to make some sort of batter. And RCA would be our kitchen assistant, with RCA alpha and beta being assistants A and B. Assistant B is always present in our kitchen to help make sure that our mixer is turned on at the appropriate times. But picture this, things are heating up. Our kitchen is on fire. Now, Assistant B is still there, but they're not great at managing this kind of stress. This is where Assistant A comes in. Assistant A is cool under stress and able to help manage our mixer even if our kitchen is on fire. Through our research, we learned how important RCA beta is for plants to survive, because without it, rubisco will not turn on, so our plants will not be able to perform photosynthesis. Also, we learned that RCA alpha is really important for helping plants manage these higher temperatures. And having this understanding of how plants function is really important in the future so we can help develop plant varieties that are able to survive and thrive at these higher temperatures so we can continue to feed the world. Thank you. Are there any questions? Yes. In the back behind you.
What happens if you take assistant A out of the plant? What happens to the plant? Does it survive fire or not?
So what we're working on right now shows us that without assistant A, our plants can still survive at these higher temperatures. They just don't do as well. So they're not able to have the same levels of photosynthesis as plants that do have RCA alpha with them. So they tend to be slightly shorter, less productive, you could say.
All the time.
Yeah, that's a great question, actually. That's one of our future directions in a sense. So assistant A actually isn't present in as much of a capacity, so there's not as much of it around. And with our plants, we also know that assistant A is not able to completely replace assistant B. So without assistant B present at all, our plant does not activate rubisco, so it performs very little to no photosynthesis.
I wonder how much temperature increase can the plant tolerate?
Yeah, that's a good question. We actually haven't tested the limits of how hot our plant can be put in. When we put our plants under heat stress, we put them at 40 degrees Celsius, which is pretty hot. So I don't know how much that is in Fahrenheit for those of us that know temperatures in Fahrenheit. Yeah. But we usually compare 25 degrees Celsius versus 40 degrees Celsius.
The final person presenting today is Guillermo Jimenez Alman. He got his PhD at the Max Planck Institute for Chemical Ecology. He's working in the lab with Fei Wei Li and George Zander. The scientific title of this presentation would be Characterizing the cannabinoid biosynthesis pathway in liverwort. Flash science presentation is shedding light on the dark side of the cannabinoids. About him, he's lived and worked in 4 countries. Owns a food truck in Ithaca. Okay.
Coming soon to Ithaca.
And equally interesting, used to teach salsa.
Wow, that's more predictable, I guess. So welcome everybody. Are you ready to get high? I mean, with food and drinks afterwards, not this. So we work with cannabinoids. We want to understand how those compounds work. Cannabinoids are basically any chemicals that can interact with the cannabinoid system of humans. And we produce them, so-called endocannabinoids, but plants produce their own, called phytocannabinoids, very well known cannabis, and they can use building blocks. Many of us like Lego, let's say, and plants like to do that. They reuse common building blocks to build complex structures. Sometimes are very similar. And cannabis is very good at doing these very well-known THC and CBD. You can see it, especially in states like New York. You can find it basically everywhere. And cannabis is very good at doing this and use complex enzymes and to produce in very high yields those compounds. That's what is the most widely, basically the only cultivar, so a crop that is used for production of these compounds. We are interested on this little known moss-like. It's not a moss, so it's a liverwort, but let's call it moss-like. At least to me, looks like a moss, and then produces this type of cannabinoid. It's not a 6. Let's say it's a 9. It's very similar. It's almost a copycat, just a stereochemistry. So the 3D structure is quite similar to the cannabinoid from cannabis. But the downside is that the production is really low. So it's like small 9. So what we want to do is to bring the workers, so the enzymes from this plant into a cousin is a moss-like 2, let's call it like this, although it's another liverwort, not a moss. So, and then engineer this in a way, because this moss-like 2 has the building blocks, but cannot connect the dots. It cannot make the cannabinoid, which come from 2 different set of Legos, let's say, cannot be connected. So we want to bring the builders in that plant and then create the cannabinoids. These new chemistries is not like a 9, it could be an A, a 7. So by doing this, we will use the whole potential of this new plant. And why do we want to do that? So the cannabinoid have been studied because of the downsides. If you look at the studies, most people are researching on the downside of cannabinoids. But we want to change that, because in a study in 2018 that showed that these new cannabinoids, let's call it like that, they keep these medicinal properties, but they don't have that strong side effects. And this is very promising if we want to exploit this further. So that's what we do. And let's go for dinner, lunch. I don't know what is this. Wow, it's still early, but yeah, dinner. Okay, thank you. Yeah, hungry.
So how do you know the medicinal effects of moss-like 2 has?
Oh, this study in 2018 showed they tried in mice, and they resemble more like the endocannabinoids, the cannabinoids that we produce. This phytocannabinoid for this liverwort then is kind of mimicking the effect, is anti-inflammatory effect and so on. That is what is observed with our endocannabinoid that are mostly derived from fatty acid like arachidonic and so on. Yeah, and on the other side, the cannabinoid from cannabis, they have the inflammatory, and then they have a lot of downsides. But yeah, let's see. So the problem is we need more of that. Yeah.
So how widely distributed are phytocannabinoids across the kingdom?
This is a good question for a real biologist. Now, there are around at least 6 other species that produce them. But the interesting thing is that I need to get a bit more technical here. Sorry about that. But cannabinoids, THC, compared to other cannabinoids, is 2 different set of building blocks which are connected, and then you get the THC, let's say that. So, but almost any other plant can do that so closely to THC and CBD. This moss-like is basically a copycat. The only difference is a stereo center and a side chain that is being used from a different pathway. So it's apparently short, it needs less engineers to do that. It's straightforward to engineer, and we hope that it's easy to engineer, not only in the moss-like 2, but also in crop plants that can do more, grow faster and so on. But this is next step. Probably bentamiana we want to use for that and start from there. Yeah.
The judges are going to repair to the judging chambers, wherever that may be, and come up with their selections. And I think there's some food out front for the rest of us for right now. I want to thank you all for coming. It's very generous of you. And you were here for the centennial edition of Flash Science at Boyce Thompson Institute. Thank you.